How are we starting out? All right, well, I'm Dylan Crossan.
I'm Marilyn Detillian.
I'm Hannah Arzagian.
And I'm Jennifer Anderson.
And we did, I guess, our project was on cancer cell dividing. And we did, I guess our article was on implementing art into STEM. And so it went from STEM to STEAM.
So we talked about the different steps of STEM and how we can directly integrate that to art. So science, we're obviously talking about a cancer cell here and just the different aspects of it dividing. And technology, we use digital imagery of micro images to create this, which we have a picture in the back. As far as engineering goes, we use the design process found in the article that we read, like Dylan already talked about, to structure our process. And for math, we talked about proportions and scale of our image in relation to our project. So the first step in engineering design process is to identify and meet our problem. Our problem was to create or reproduce an image from a micro image that we found into an artwork. So we abstracted it to do that. The second part is we needed to research the problem, and we did that by understanding more about batiks and how to use that paint on the silk.
The next steps in the design process are developing possible solutions and then also selecting the possible solution. And so for us, we had not only to choose a micro image that we felt was appropriate for teaching both art and science, but also deciding on a technique to use. And as she mentioned, we used batik within our constraints that we had present for us. And that was what we were dealing with then, our cancer cells dividing and use of batik silk painting for how we're going to display the information.
Then step 5 was construct a prototype, and I guess in order to do that, we drew, I guess, that picture out onto paper, and we kind of scaled it out and put it on the canvas for the final project. And another type of prototype I guess we kind of thought about was when we actually did our small sample ones. It kind of gave us a feel of how we're going to do it. Step 6 is to test and evaluate the solutions, and basically that's just throwing the paint onto the canvas and seeing how it turns out. And that's pretty much step 6. So.
Step 7 was communicate the solution, which after we threw the paint on the canvas, then we decided how we're going to change it, how we're going to communicate that maybe to a client or something. We communicated it right on to here with the different colors, and this is our visual representation of cell division. The last step is step 8, which is redesign, which this is kind of a mock-up or experiment or prototype, and we're going to redesign next time. We'll see what we used and what experiments worked and what errors we had, and we'll integrate that all into a new project to redesign it.
And throughout this process, we talked specifically about the art content and the science content. And so for art, we really explored visual texture on this piece of cloth, and just how we see things as having texture, even though this whole thing is very smooth. We also talked about color and complementary colors. Our images, to start out with, had blue and orange, which are complements. So automatically that just really makes it pop. So we wanted to integrate that into our design. We used contour lines for the wax resist to outline the main aspects of our shape. And then tints and shades were also explored with vinegar and alcohol in these small dots down here.
As far as science content goes, we could go on, or we can go on to discuss cell division. This is a cancer cell speaking to mitosis, the actual replication of a cell. And
I guess apoptosis as well as